The human experiences of patients, the workforce, and ecosystem partners are interrelated and directly affect business outcomes. At Allsteel, we believe in contributing and investing in workplace strategy and product solutions to an industry with a hand in the health and safety of our global population. It is highly critical through our design applications and product development to encourage and support the best work of teams involved. Thoughtful planning, flexible solutions, and innovative partnerships create the right landscape to allow each employee, scientist, and administrator to produce their best result. Within the life sciences industry, we are designing environments that have the ability to scale spaces as your businesses change and transition teams from trial phases through development and launch that can create critical business efficiencies and support talent to achieve their fullest potential. At Allsteel, we transform workplaces for the better in a collaborative and human-centric way by designing for real people and their ambitions. Allsteel is designed to do more.
Good morning. My name is Todd Kilbaugh. I am an anesthesiologist and an intensive care physician at the Children's Hospital of Philadelphia. I'm also an endowed chair of translational science and innovation at the Children's Hospital of Philadelphia and University of Pennsylvania. It's my pleasure. I get to just introduce some very lovely people. It was actually interesting to hear John, the previous speaker, talk about Emily Whitehead, the first patient that received CAR T-cell therapy. Interestingly enough, I was actually the ICU doctor that was on right after she received her therapy. And in those days, the early moments of CAR T therapy caused massive inflammatory reaction. She actually arrested, and I was the one that coded her and then brought her to the ICU and then took care of her for a week. So it's amazing how all these things sort of come around, and I haven't really thought about that in a long time. I didn't even know that she was a freshman at Penn. So it's amazing that ecosystems sort of help each other, how as people as in the real estate industry, people in pharma, people in academics, how we as a community are really saving patients' lives. And no one knows it more than myself and some of my partners here that I do research with. Meeson Wang, who is a radiologist. All their bios are in there, so I won't go through all of it. But she's an amazing woman, mother of 3, works harder than anybody I've ever met, and just does incredible science. And beyond that, she's just an incredible role model and person. Bill Parentas, I've known for a long time now. He's an attending surgeon at the Children's Hospital of Philadelphia, fetal surgeon, is one of the leaders in the fetal group from a clinical standpoint. Just received a $25 million grant from the NIH. Is looking at me like, don't embarrass me. It's like my 9 or 11-year-old looking at me. But pretty amazing stuff. Those type of dollars don't fly out of the NIH and those big, large sums unless you're pretty amazing and impressive. And John, I just met, John am I saying it right? Karanakolis is a professor of molecular therapies at the Fox Chase Center, and even though we just met, I'm sure he's just as fantastic. So each is going to give an 8 to 10 minute presentation. We'll hold questions, and then we'll leave about 10 minutes at the end. I'm just up here for eye candy, and I'll let them take it away. Ready? I think, Bill, you're up first, yeah?
Who's first?
Let's see. That's you.
Good morning, everybody. Thanks, Todd. So it's a pleasure to talk to you today about an area of research and interest that we're involved in. So 1 in 33 babies is born every day with a birth defect. It costs our healthcare system billions of dollars every year. And within this category of birth defects, the World Health Organization estimates that up to 7,000 to 10,000 of these diseases can be attributed to mutations or abnormalities in single genes or monogenic birth defects. Now, we have had significant advances in medical technology to the point that we can now diagnose most structural as well as genetic birth defects before a baby is even born. Structural birth defects, I mean like tumors growing off of the tailbone or a hole in the diaphragm. Many of these defects cause significant morbidity or healthcare problems and even death to a fetus before or immediately after they're born. And so the ability to diagnose these problems, combined with the fact that they cause significant health problems before birth, obviously begs the question of, can you treat them before birth? And so the idea of open fetal surgery for a structural or an anatomic birth defect is not new. It grew out of UCSF in the 80s, and now at CHOP, the Children's Hospital of Philadelphia, we perform as a huge team on average of one fetal surgery every 1 to 2 weeks. And so if I can get this to work, it is a real surgery, so if you're a little squeamish, maybe don't look, but we're just going to show you. So this is a fetal surgery for a tumor growing off the tailbone. It's a big team led by my mentor, Dr. Adzik. And here you can see the lower half of the fetus at 24 weeks gestation being delivered from the uterus. The upper half is kept inside. You can see a big tumor growing off of the base or the bottom of the fetus. And again, as part of the big team, what we do is we just cut off that external part of the tumor because it's highly vascular. And it's almost as big as the baby at this point. And so the heart is under a tremendous amount of strain, and the fetus is going into heart failure because the heart is working hard enough to provide blood to the tumor as well as provide blood to the baby. So we use a bunch of special instruments to control bleeding. We debulk or resect that external part of the tumor. We oversew the skin where the tumor had come from. And then we return the lower half of the fetus back into the uterus, seen here, and we close the uterus up. The mother, at this point, again, was 24 weeks gestation. The uterus is returned into the abdomen, and then the goal is to keep the mom pregnant as long as possible, ideally until term or 40 weeks gestation. And so I show you this video for 2 reasons. One, because you don't get to see this every day, so it's nice to share with you. And then 2, just to highlight that that what we're going to talk about and what we're interested in maybe isn't that crazy of an idea if we're doing this already for structural birth defects. And so what we think the future of fetal interventions or prenatal medicine is a more precision fetal medicine using gene editing technology that Dr. Epstein spoke about, where with the mother awake on a table and using an ultrasound, you can direct a needle into the umbilical vessels and deliver your gene editing technology to the organ of interest, here the lung, and correct the underlying mutation in the gene to fix the disease before the baby is even born. And so this is what we study in my lab in collaboration with people from Penn and across the country. So we use CRISPR-Cas9 gene editing technology. I'm not going to go into the details of this, but these were developed by people out of MIT and Berkeley. People won the Nobel Prize for this. But the bottom line is you can use this therapy to target specific sites in the DNA and make very precise changes to the DNA to correct a missequence that's causing a disease into what the sequence should be. So people are doing this after birth, and so the question is, why would you ever want to do this before birth? And the idea is just to take advantage of the normal developmental properties of the fetus to institute the therapy in a potentially more efficient and a safer manner. And so what are some of these properties? Well, the fetus is small, and a lot of these therapies are dosed based on the weight of the recipient, and so you can maximize the therapeutic dose. And then from a practical consideration, it's very expensive and very tedious to make GMP quality drug products. And so if you're making a smaller amount of the drug product, it obviously offloads a lot of the burden of making that drug product. There are some barriers that exist after birth, like a blood-brain barrier or the barrier in our lungs that we evolutionarily developed to try to prevent infections from going into our brain or going into our lungs. We're using viral particles to deliver this therapy, and so it would only make sense that it's hard to target those organs after birth. Before birth, those robust barriers aren't as present. And the immune system of the fetus isn't as mature, and so the fact that a lot of the therapies that we're delivering are delivered by viral vectors and they're bacterial-based. If you do it in a fetus, it's less likely to be rejected by the immune system. And then finally, I would argue that if you're correcting a disease-causing problem, you want to correct it in a stem cell or a progenitor cell so that whatever you correct goes on for the rest of the life of the patient. And these stem and progenitor cells are more accessible in a fetus. And then this all comes together for the ideal vision in that there are some diseases where the pathology starts before birth, and once the baby is born, it's hard to turn back the hands of time and reverse that pathology. So the ultimate goal is to provide a single, one-time cure before the onset of irreversible pathologies. So to address this, our lab kind of focuses on two main buckets. The first is which diseases do we want to target, and the second is what are developing delivery approaches to target the organs of interest with the ultimate goal of clinical translation. This is essentially the workflow that we follow in the lab. We do initial studies in mouse models of diseases, as well as human cells and cell culture. And then once we've demonstrated some feasibility in these models, we move them into humanized mouse models. So these are mice that we create that contain the human gene and the disease that you're interested in, because the way the editing technology works is that it's sequence specific. And so our sequence is different than a mouse sequence. So you need to make these humanized models. And then ultimately we progress onto large animal models, including non-human primates and fetal sheep models, always evaluating efficacy and safety in these models. These are a couple of the top diseases that we're interested in: lysosomal storage diseases, which cause a lot of neurodegenerative problems before a baby is born; genetic lung diseases like cystic fibrosis. It's obvious. Before birth, you don't need your lungs to breathe, but as soon as you're born, your lungs are critical. And so if you have a genetic lung disease, you can have significant problems at the time of birth. Epidermolysis bullosa is a skin disease, and just through the process of being born, babies can lose skin or patches of skin. It predisposes them to skin cancer and infections throughout life. We're also interested in Friedreich's ataxia, a neurodegenerative disease, and some hemoglobinopathies such as alpha thalassemia. And then the next 2 to 3 slides is the most science I'm going to show you. So we've been able to study in mouse models of a metabolic liver disease, as well as in a mouse model of a genetic lung disease, that we can rescue the disease phenotype. So in these models, the mice die soon after birth. By applying the gene editing therapies before birth, the mice survive and live a normal life. This is one of the main diseases that we're interested in. It's Hurler syndrome. It's a lysosomal storage disease. It's a prevalence of about 1 in 100,000, and it is known that the pathology, especially the brain pathology, starts before a baby is even born. There are some therapies out for it, but unfortunately they don't work very well, and they have toxicities associated with it. And despite some of these therapies, a lot of patients will die in their teenage years because of it. In collaboration, again, with colleagues from Penn Bioengineering and the Penn Genetics team, we've developed some lipid nanoparticles that could more robustly and efficiently target the brain. And then in following our workflow, we first did these studies in mouse models, showing that we could correct the disease phenotype in the mouse. We then took our product and we sent it to a good collaborator and friend we have in Singapore, and we did studies to show that this delivery technique could target the brain in a non-human primate fetus. And then finally, because of our association with CHOP, we have access to patient samples. And I think that's one of the highlights of the Philadelphia area, is that not only do we have the basic research, but we have all these robust hospital systems that facilitate the translation of this science from the bench to the bedside. And using these patient samples, we were able to show that we could use this delivery approach to target different cells in the brain, and that it was stable in the CSF, which is the fluid in which it would be injected. And so that's a brief overview, and we like to think of this as the tip of the iceberg. These are some of the target diseases above the water that we're interested in, but once we show that it's feasible and safe, there are a number of diseases affecting patients and their families that could potentially benefit from an approach like this. And so I like to end on this. This is the picture of the first fetal family reunion at the Children's Hospital of Philadelphia back in 1996, started again by my mentor, Dr. Scott Adzik. And the joke on this picture is that the number of healthcare providers is far higher than the number of patients that were treated. And then you fast forward to our fetal family reunion back in 2 years ago, and it's gotten so big that we now moved it from CHOP to the Philadelphia Zoo, and we take it with a drone, the picture with a drone camera. And so, I like to look at this picture, and these are all for fetuses with an anatomic or a structural birth defect. And I like to dream what this picture will look like when we are also able to treat genetic birth defects, which significantly outnumber anatomic birth defects. And so this is work by a number of people, and this just lists them as well. And so thank you for your attention, and I'd be happy to take questions at the end.
Good morning, everyone. It's very exciting to be here in front of all the accomplished people here. So I'll be talking about targeted drug delivery using bubbles. I wish we had champagne to toast, but all the talk will be about bubbles. You'll be thinking about bubbles when you walk out, and you'll love bubbles after this talk. So I'm actually a pediatric radiologist and actually physician scientist at Children's Hospital of Philadelphia. I'm also an assistant professor at University of Pennsylvania. I met Todd ever since I joined CHOP, and he's just an amazing, crazier than me, but just a fantastic person, and I'm happy that you invited me to talk here. And I also work with Bill on a lot of projects, and he is just so amazing. I'm just so humbled to be in this environment. So these are my disclosures, some of the funding sources and the people I work with. So I'd like to talk about kind of the current landscape. What have we been doing with drugs in general? So if you look around, a lot of the drugs are systemic delivery. So either you take it orally or you inject systemically. And it's associated with a lot of issues. There's systemic toxicities associated with it, and there's low yield at the target organ. If you can imagine, you're taking orally, some of it will be, not going to the target. So this is, as you're familiar, this is a big company, AstraZeneca. It's in the front page, and they're devoting a lot of efforts in targeted delivery technologies. And in this slide, they're talking about lipid nanoparticles. And this is what Bill has been talking about, and a lot of efforts are going here. Why? Because if you just inject it, it's hard to go to target organ. But lipid nanoparticles can encapsulate drugs. Now, the LNP market size is projected to be over $700 million by 2028. Now, what's the issue of nano platforms? Well, we just talked about targeting to brain. Now we're going to talk about targeting to tumor. We're going to talk about targeting to bowel. A lot of these targeting needs sophisticated functionalization of nanoparticle itself. You have to understand the chemical milieu. You have to understand how the barrier works and how it's going to interact with the barrier and enter the target organ. So as you can imagine, AstraZeneca and other companies working on this, devoting tremendous effort, human resources, chemistry, biology, into understanding how to develop each and every nanoparticle to reach the target organ. It also is an issue because nanoparticles get uptaken by the liver a lot, and they don't go, their yield is a little bit lower than you would imagine. So what we're working on is microbubbles. So nano is nanoscale, microbubbles is micron size. These are 2 to 3 micron size. It can be a little bit smaller, it can be bigger. But microbubbles are basically phospholipid layer, or it could be any shell that has either gas in it, or it can contain any other therapeutics. Now this is used as clinical contrast agents, believe it or not. It's an ultrasound contrast agent. They contain gas, and so they oscillate under ultrasound pressure, and they reflect sound waves. Ultrasound is all about sound. So you shoot a sound, and they oscillate, they shoot back the sound, and we're just listening. And so this is actually FDA-approved ultrasound contrast agent used in pediatric patients and also adults. Now, what can you do with it? Well, it can encapsulate a lot of things, including genes, proteins, gas, LNPs, drugs. And what's the benefit of it? Well, they stay in the circulation for a long time. It can stay for max 10 minutes, maybe more. It depends on the stability of the shell. Now, what happens if you apply an ultrasound pulse? Remember, it's responding to the sound wave. And so when you're shooting a low energy pulse, it just oscillates. It gives off the sound back. But if you shoot a high pulse, which I'm not talking about a really dangerous pulse, it's a pulse that is equivalent to grayscale ultrasound, just a conventional ultrasound, then it kind of pops. And when it pops at a target site, you can pop it at carotid, then it releases the factors. So the point is, it's a ship. It's a ship that carries things, and you don't want to deliver it to all organs because it's toxic, and the yield at the therapeutic target organ is low. So what you're virtually doing is you're carrying in a ship the cargo that you want, and you're delivering to the target site when you want to, with a pulse that is not that dangerous. So this is just another demonstration of what can happen. So you have a bubble with a cargo loaded. You can load it inside, you can load it in the shell, you can load it at the surface of the shell. There are various methods of manufacturing this. And as you shoot an ultrasound pulse, it oscillates and cavitates if it's a high enough energy, again, which is not dangerous. It's a grayscale, conventional energy that we're talking about. Now, those two brain MRI images are of pigs that I've done in collaboration with Dr. Kilbach here. And they're treated with traumatic brain injury, so they undergo contusion, focal contusion. On the left is day 1, so you can see the white signal is the edema, and the black is the hemorrhage contusion site. You can see the marked edema. Now, we put xenon into the bubbles. We intravenously administered just a couple minutes, and that's day 5. Edema shrank. Now, I'm not going to go into detail about the xenon itself. It's a very interesting anesthetic agent, but it's neuroprotective, and we did some studies showing that it does reduce edema. We're testing other therapeutic agents in a variety of brain injury models in pigs. But this goes to show that targeted release of drugs can be effective, and especially in this disease where there's no treatment at all. Hopefully this video plays. Yes. So these are bubbles, and those sticking out, those can be antibodies. So these bubbles can also be targeted, just like the nanos can be targeted, but they're stable. And they go through the circulation. They can contain xenon gas in this case, or they can contain genes, drugs, any therapeutic agents of your choice. It can contain RNAs. And you would be administering through an IV, peripheral IV, just the way you would administer any other drug contrast or drug agents. And as it goes, you see the bubbles are smaller than red blood cells. They are a couple of microns in size, and they are stable, and that's the that's the manufacturing part. You have to make sure that they're stable until you shoot an ultrasound pulse, in this case brain, and then you can actually pop it and deliver the drugs so that you're not releasing all the drugs in all over the place so as to create high systemic toxicity, which is a major issue in some of the promising clinical trials, including genes and CAR-T. We have had, deaths related to the systemic toxicity, so there is a tremendous need for targeted drug delivery in this space. And there it's just showing that the released xenon is serving as a neuroprotective agent in the brain. Now, interestingly, I just told you that the bubbles can be contrast agents, and it can be. So that is on the right, you see half of the brain. That's a half of the brain in coronal plane of a pig. We can depict microcirculation at the less than 10 microns in resolution. Why? Because we can see these bubbles, we can track these bubbles, we can exactly know where they're going before we pop it. And that's the beauty of it, and it's just another video showing that, it by oscillating can give off sound waves, and that is actually what we see, and we can resolve into super resolution microvascular imaging. That's another field of study in my lab that we're doing in collaboration. And this is just showing that, if you resolve using complex computing processes, you can see that the pig retina, that's a pig retina. You can see under less than 10 microns in resolution. Remember, MRI resolution is 1 millimeter. So this is 100-fold higher. And that's a human aorta, human heart. We can track all these 4D flows using these bubbles, meaning that it not only serves as a ship that carries all the drugs to targeted sites, but we can see these as we're delivering, and we can actually measure how much we're delivering, of course, with some validation in the lab. So in the future, I think that there is a lot of potential to utilize these microbubble platforms as a versatile, cost-effective, and safe ship for organ or site-specific delivery and treatment. Thank you so much.
Thank you so much. Well, it's a joy to be here today. My name is John Kerr-Nicholas. I'm a professor at Fox Chase Cancer Center and at Temple University. The thing that I'm really passionate about is designing new things on the computer, so specifically proteins and small molecules that can bind to proteins or other proteins that can bind to them, designing these things on the computer and then building them in the lab and seeing if they really work as we design them. And so this movie that you're seeing on the left here, this is a little bit, it's nostalgic for me. This is something that I designed about 20 years ago. This is a pair of proteins that were supposed to interact with one another. And this was designed 20 years ago on the computer when many of these algorithms were really in their nascency. And then we built them in the lab and figured out how they worked and showed that they interacted with one another precisely the way that we had intended them to on the computer. So by way of disclosures, because I'm in academia, it's traditional to start with the disclosure slide. So I'm involved with a few companies, and my lab is involved with a few companies, and none of these are based out of Philly. And that's sort of a shame. And that's something that I think is changing, and I think there is a groundswell of momentum for more and more innovation and more commercialization of laboratory discoveries that's happening and staying in Philadelphia, and I'm really excited about that. So the internet, of course, has its own culture. One of the sort of long-standing debates on the internet is whether giraffes, if they were to wear hats, would wear one hat or two hats. And so for those of you that don't know what I'm talking about, I guess the reason I bring it up is if it were me, I would be wearing two hats because that's what I think giraffes would wear. And that is what I wear, in fact. And so I alluded to my roles. And so I'm a professor at Fox Chase Cancer Center here in Philadelphia, where I lead our cancer signaling and microenvironment program. And then in parallel, I'm director of the Mulder Center for Drug Discovery Research in the Temple University School of Pharmacy. And so on the Fox Chase side, really what we have at Fox Chase, and I'm going to delve into this a little bit more, is a lot of expertise in cancer biology and in thinking about new potential targets for intervention in cancer. And what we have in the Temple Pharmacy School is a lot of expertise in chemistry and medicinal chemistry and drug discovery. And so these two together really synergize in a way that I'm going to describe for you now. So for those of you that don't know Fox Chase as well as I do, Fox Chase was founded in 1904. It was one of the nation's first dedicated cancer hospitals. And in 1974, the NCI decided to have a series of dedicated cancer centers or comprehensive cancer centers throughout the nation, and Fox Chase was among the first of these. Fox Chase, the researchers at Fox Chase have included Nobel laureates, including Barry Blumberg and Ernie Rose. And so if you sort of look at the spacing of these, we're about due for a third Nobel laureate at Fox Chase. That was a bold statement. The research at Fox Chase has really included absolutely seminal laboratory discoveries in the field of cancer and our understanding of cancer. And so famously among these is identification of the Philadelphia chromosome, a gene translocation that underlies CLL, chronic myeloid leukemia, CML. But in addition to this, there's been a number of key investigations in the laboratory at Fox Chase that have identified drivers of cancer, and many of these in turn have spurred discovery and development of cancer drugs that are used every day today. And so the mission statement of Fox Chase is to prevail over cancer, marshaling heart and mind in bold scientific discovery, pioneering prevention, and compassionate care. And the reason I show that is because I think it really does encapsulate exactly who we are at Fox Chase and what we're doing. And so now to shift gears slightly, let me tell you about the Mulder Center, which is much newer. It was established in 2009 with a donation from alumni of the Temple Pharmacy School, Lonnie and Sharon Mulder. And then it was boosted by a second pledge by the two of them in 2015. I started as director of the Mulder Center in 2022. And I alluded to sort of my own focus and my own passion of designing things on the computer and then building them and testing them. And so this is part of what I brought to the Mulder Center, is an emphasis on AI-driven drug discovery to complement a lot of the existing strengths in medicinal chemistry and pharmacology and PK, pharmacokinetics in this case. So the goal of the Mulder Center is to enable drug discovery research at Temple and in the Philadelphia research community by providing access to expertise and collaborative services. And so what we have in the Temple Pharmacy School is experts in various components, various elements of the drug discovery process that we can bring to collaborative projects between the Mulder Center as well as partners both in academia as well as biotech startups in the Philadelphia area. And so I'm going to show you a couple data slides that aren't mine. And so essentially one of the things that I'm tasked with in these dual roles at Fox Chase and at Temple is in thinking about what should be the role of academic drug discovery. Because pharmaceutical companies are playing a very large and distinct role in the ecosystem that brings drugs to patients. Biotechs play an absolutely indispensable role as well that's different and non-overlapping from big pharma. And so what should be the role of academic drug discovery? And that's something that I think about a lot. And so in 2014, the NIH commissioned a group called the Illuminating the Druggable Genome, which was essentially tasked with figuring out among the proteome, among all of the human proteins, all the genes expressed in human cells, which are the ones that make for good drug targets? Where should we be directing our attention? And so this commission tasked by NIH with this job worked hard on it for several years. And so here's a little bit of a technical slide, but what I want you to see is the blue bars. So along the horizontal axis are different classes of proteins, different types of proteins in cells. And so the blue bars are the targets for which we have drugs currently in the clinic. The green bars are cases where we have drugs that are working their way towards the clinic. They've been shown to work in mice or they're undergoing clinical trials currently. And the red bars are genes or protein targets for which we know unambiguously that they are important drivers of disease, and yet we have nothing. We have no drugs to target any of these proteins, these genes that are in the red bars. And so the text is a little bit small, but the middle ones that you can see where the green and blue bars are very high are G protein coupled receptors, ion channels, kinases, nuclear receptors. So to the people that think about drug discovery in the audience, these are the well-trod targets. These are the cases where when one such target is identified and implicated in disease, we sort of know that we can develop a drug for it. I mean, they're targets that are considered highly druggable. But then all the stuff on the right part of this slide, you can see how big those orange bars are. These are genes that are unequivocally implicated as disease-causative genes, and yet we don't have small molecules, we don't have drugs to target these. And so that's precisely what we've been doing through a new initiative, a partnership between Fox Chase Cancer Center and the Mulder Center. And so this is a series of projects that are specifically designed to go after some of these high-hanging fruits in drug discovery, these targets that we know are very important for disease, and yet we don't have drugs to address them yet. And so some of them are things like epigenetic modulators, playing to strengths at Fox Chase Cancer Center, but then a lot of these are sort of mechanistically subtle things that require exquisite precision medicine that come from the computational AI-driven strengths of the Mulder Center. So things like a kinase that we know does two things in cells, and we want to block one of them but not the other. Or small molecules that degrade their target of interest, such as these are called PROTACs or molecular glues. And so using some of these new therapeutic modalities, we can go after some of these proteins that we know really well are disease causative, but we don't have traditional tools of drug discovery have sort of let us down in targeting these challenging targets. And so the prime opportunities for academic drug discovery research really come to addressing this other category, the ability to reach into what would be considered riskier territory for large pharmaceutical companies or even for biotechs, and de-risking these by identifying starting points for further drug development. And then to address this other category, given the challenges associated with these whole target classes, is developing new therapeutic modalities. And so you heard earlier about RNA therapies and CAR Ts developed at Penn, and those are precisely the types of new therapeutic modalities discovered and developed initially in academia that became absolutely essential translational tools, these new therapeutic modalities. And so increasingly, development of new therapeutic modalities also underlies a lot of the collaborative research between Fox Chase Cancer Center and the Mulder Center. And so here, this one example that I'm showing you is building antibodies that can be activated using small molecules. And so it's sort of combining the best of both worlds to address targets that wouldn't be addressable in other ways. Thank you very much, and I'm excited for our panel discussion ahead.
We have a few minutes for some questions for our presenters. Please feel free to ask any questions. Go ahead.
So I'm curious about the microbubbles and the delivery. In the case of like gene therapy, so like for viral gene therapy, we're going to take a pill that you have to inject it into a specific site. So how do you target a specific organ with the microbubbles?
Can you hear?
Okay. The question was the ability, how do you target specific organs in relation to gene therapy with the microbubbles?
Yes, so that's the advantage that microbubbles have, is that you don't have to functionalize for targeting. It would go systemically, and wherever you want it to be delivered to and pop, that's where you apply the ultrasound pulse. So the system, yeah, exactly. It's not even guiding, it's just, yes, guided release. So it will circulate everywhere. You formulate it so that it's stable in the circulation for a long time until you apply the ultrasound pulse. Remember, all the organs have supplying vessels. And so in the brain, for instance, it would be carotid. So if you're popping at the carotid, you would just be releasing at the brain. Yes, that's a great question.
Hi, thank you again for your all very interesting. My name is Krista. To piggyback off of your question, I'm curious about what happens or how the body rids the microbubbles after the popping.
Sure. So the bubbles that are used in the clinical studies, they're cleared through the liver. So the shells are very safe. They're biologically inert and safe, phospholipid monolayer, and they're uptaken by the macrophages in the liver, and they're cleared away. Thank you.
Hi, thank you. Dave Garby. Question for William. The diseases that you're trying to treat, are they monogenetic, or are we also trying to find ways to treat diseases that we know maybe are, caused by multiple genes?
Yeah, it's a great question. So I think that for gene editing strategies, the diseases are monogenic. So a single gene, depending on the approach, ideally there's a common mutation that affects the majority of patients that have that disease. So you could develop one gene editing therapy for that common mutation in that single gene. You could imagine if there are 100 different mutations, developing 100 different gene editing therapies and getting that through the FDA is very difficult, right? And so ideally it's a monogenic disease with a common mutation. Or alternatively, an endogenous gene that you can silence or inactivate that can treat the disease independent of the disease-causing mutation.
Go ahead.
Hi, I had a question about the fetal surgery. I didn't hear anything about the moms. Like, is there any social support or like the mom understands what's happening to her body? And as a mom myself, like, I know you said the patient, but isn't the mom also a patient? Is there any sort of after surgery care for her, because you're splitting her open uterus and abdominal wall? Is there some parallel support, medical and maybe social support, so that she also recovers while the baby that she thought was perfect is also recovering? Is there something for that as well? Thank you.
Yeah, thank you for that question. That's excellent, and thank you for bringing that up. So at CHOP and many of the other fetal centers around the country, which are only a handful, there is a huge network of team members to take care of the mom and the fetus. Our guiding principle is that the mom is the most important patient in any fetal surgery, right? And so we we have a minimum of a 2-day multidisciplinary counseling session where they see maternal fetal medicine doctors, pediatric surgeons, social work. We have Joanna Cole, who's a psychologist that works just within the maternal fetal medicine center to speak with the mother, both before the surgery as well as long-term after the surgery. But it is an excellent point, and is part of the risks of fetal surgery. It's why there are very stringent selection criteria as to which diseases would be potentially amenable to a fetal intervention, right? And similarly for genetic stuff, we're not proposing that every genetic disease would be amenable to a prenatal intervention, right? We need to pick the disease and the family that would benefit from the most, knowing that there's risks to both the mother and the fetus, with the health of the mother being the ultimate, both the mental as well as the physical health of the mother being the ultimate priority.
Good morning, everyone. Dr. Tia Lyles-Williams. This question, I guess, is kind of for all of you guys at CHOP. 2 questions. I noticed the picture with the fetal patients and their respective mothers, and wasn't a lot of diversity there, right? And so we're in heavy conversation about health equity and removing health disparities, and there's 2 ways of doing that, right? One, there's the aspect of making sure that we include Black and brown in these opportunities and explicitly pursuing those community members at hospitals where they don't have insurance, they may not have access, but they potentially and most likely will fit the mold or the requirements for that particular study. So that's one question, how are you guys addressing that? And then the second thing, I think Mr. Karankolas, hope I pronounced your name correctly, when you talked about, about startups and looking at and investigating what we consider non-druggable assets today, are variants, genetic variants, be included in their research, because oftentimes Black and brown don't necessarily fit the exact protocol. Most times they actually get treated, for example, in 1 round versus 2 rounds, but in research they're often documented as outliers, and no further investigation goes as to why their treatment was more successful or less successful per those protocol expectations, hence why we're just now getting a sickle cell therapy via Global Blood Therapeutics and their emergent acquisition with Gilead. So what are the, I guess you could say, the thoughts or the strategies of going down that route of not just looking at non-druggable assets, but those variants and outliers that we call in Black and brown patients today?
Do you want to go first?
Sure, I'll go first. So I think it's a very critical point that you make. So, and I think a lot of it has to do with outreach. So we are focused on treating and offering, it's not just treating, it's actually counseling, right? So the biggest benefit of a fetal center is not doing the fetal surgery, because for all the families that we see, it's only a small percentage that actually get a fetal procedure. The real benefit is hooking them into a healthcare system, making sure that the mother and the fetus know what is going on, so that after the baby is born, they can get appropriate care. And so the biggest goal is outreach, and we make a concerted effort to spread the word and make people aware of the services that we have. We've treated patients from all 50 states and from over 20 countries based on this, this principle. And due to philanthropic donations from the Garbos family as well as others, we have funds available to offset the cost of travel. We work closely with the Ronald McDonald House as well as local hotels to offset and even pay for housing for families that can't afford to come to CHOP to receive the care. And so, and I don't have the data in terms of the diversity of the patient population that we treat. I can tell you that it is not specifically limited to one population or the other, but I think we always need to do a better job at making sure that this type of healthcare, which is complicated, is available to every patient that wants it.
So from my side, I'm going to give you 2 answers, if I may, for different parts. So you brought up diversity in clinical trials, and this is, so the lack of, so it's unambiguous that there's a lack of diversity in clinical trials. And I think the problem, and the reason I'm going to give you 2 answers is that I think, as with other cases where there's pervasive and systemic inequities, I think it's every step along the way contributes. And so with respect to clinical trials, lack of diversity in clinical trials comes from both lack of participation in clinical trials because of who's coming to the centers and receiving care. And so in part, we have a new program called ADEPT, which is broadening participation in clinical trials, which is centered at the Broad Street Temple Health Hospital, which is explicitly designed to have broader and more diverse inclusion in clinical trials. Then the earlier part of the pipeline, though, and you brought up variants, and that's a really important point, is that from a very basic science standpoint, we do a lot of work on cancer cell lines, so cells that have been, with patient permission, donated to use in the course of research. And the cells that we're carrying out the very basic science studies on are primarily coming from white men. And that's a problem because when we test our drug candidates to evaluate them at the very, very earliest stages of drug discovery, we're testing them on cells that come from white men. And so we're steering which drug candidates we advance based on information that's inherently not representative of the broader population that these drugs are meant to help. And so I bring this up because it's not in clinical trials, it's at the very, very earliest stages of basic discovery. And the branch points that come there affect the efficacy of the drugs years later. And so among the efforts that are ongoing now to address this is carrying out research using cell lines that come from a much broader population so that the conclusions that we draw as we develop these drugs from the very earliest stages will be much more representative of the populations that they can benefit. And as with anything relating to health disparities, I think there's a lot of progress being made currently. We're far behind where we should be, but I think we're making up for lost time, and I think there is momentum and change is happening pretty rapidly, but it's overdue. Thank you for the excellent question.
Okay, I think that we only had 10 minutes. Those were all incredible questions. I think from the panel you can see that the change is happening rapidly in this area, whether it's Drexel or it's Fox Chase or it's CHOP or Penn or it's Thomas Jefferson. The amount of data that is coming out, the amount of data that will be more inclusive of diverse populations, the ability to do things that we're treating patients before they ever develop these disease processes, things like non-invasive procedures that will deliver drugs, things that we can use AI and other predictive models to actually design drugs that are safe and have less toxicity before we ever even hit a patient. The amount of explosion that's happening both in this region and around the world is incredible. And the real question that will then come up is, are we moving drugs ethically? Are we moving therapies ethically to diverse populations? Are we taking an approach that is sustainable over a long period of time, both from a financial standpoint and from a cultural standpoint? And I think these are all great questions and part of the fabric that's going to move forward over the next 20 to 50 years here in the city. So thank you so much.